Hi everyone, today we're going to be solving Cambridge International AS and A-Level 9700 Paper 41, May-June 2023. Figure 1.1 shows the transmission electron micrograph of a part of a chloroplast. We can see parts labeled A, B, C, D. A is the stroma, B is the inside of the thylakoid, so we'll call it thylakoid lumen, C is the thylakoid membrane, and D is the ribosome. Table 1.1 describes some function that occurs in different parts of a chloroplast. Complete the table 1.1, identifying the letter on figure 1.1 that is a location matching the description. Each letter may be used once, more than once, or not at all. So the first description says accumulates buildup of high concentration of protons. High concentration of protons is built up in thylakoid lumen. So the letter is B. Makes triose phosphate. Triose phosphate is produced in the stroma. It is in the light-independent portion of the reaction. So, it will be stroma, which is A. Makes some chloroplast protein. Proteins are made in ribosomes, and the ribosomes are labeled with D. Proton pumps. Proton pumps are present in thylakoid membrane, which is C. Membranes of the type labeled C in figure 1.1 were made into a liquid extract. Chromatography was then used to separate and identify the colored components in this extract. So, the membrane of the liquid, the labeled C, was thylakoid membrane. And we know the thylakoid membrane consists of primary pigment, which is chlorophyll, and also contains accessory pigment, which are beta carotene and carotenoids. So beta carotene and xanthophyll. The resulting chromatogram showed that these membranes contain a yellow pigment, an orange pigment, and a green-brown pigment, and two different green pigments. Describe how you would carry out chromatography to separate and identify the colored pigments in the liquid extract of C. When we are asked to describe the process of chromatography, so we need to mention how the chromatography is carried out. And finally, how we match result to identify. So the answer can be written as follows. The spots of the extract was placed on pencil line. The base of the chromatography paper was suspended in alcohol. The solvent front was marked after the solvent reached near the top and the paper was removed. RF value is equals to the distance moved by the solute divided by the distance moved by the solvent. And as a last added point we can write, then compare the RF with known RF values to identify pigments. Only 4 marking points will be needed for this particular answer. Explain why membrane C has many different colored pigments to function efficiently. So we can write membrane C consists of chlorophyll A, chlorophyll B, xanthophyll. You can also include carotenes. The pigments, they harvest light energy. With a different pigment, a range of wavelengths can be absorbed. This increases the rate of photosynthesis. The natural ecosystem on Hawatucks Island in Alaska was disrupted in the 1780s when brown rats, Rattus norvicus, swam to the island from a sinking ship, then rapidly increased their population size. The rats occupied a new niche on the island as predators. The rats ate the eggs and chicks of birds such as black oystercatcher, the scientific name Hematopus bachmani, and the glaucous-winged gull, scientific name Laurus glaucescens. These birds make nests, lay eggs, and rear their chicks on the beaches of the island. Define the term ecosystem and niche. Our ecosystem can be defined as a self-contained unit containing community of organisms and their interactions with the biotic and abiotic factors of the environment. Define the term niche. Niche can be defined as the role of a species within its ecosystem. Conservation ecologists carried out a project to try to restore the natural ecosystem of Hawatucks Island. In 2008, they removed all rats from the island. Before removing the rats, the ecologist measured the abundance of birds, invertebrates, and seaweeds on 8 of the island's beaches. Seaweeds are large algae that grows attached to rocks on the beach. The ecologists repeated the measurements in 2013 and in 19 so that they could calculate the percentage change in the abundance from 2008. To measure the abundance of invertebrates and seaweeds, the ecologists used this method. They laid 30-meter tapes from high tide mark to low tide mark on the beach. They placed quadrates at 5 meters intervals next to the tapes. They took a photograph of each quadrate. They analyzed the photograph to calculate the percentage cover of seaweed and the percentage cover of invertebrates such as mussels and sea snails. State the name of the sampling technique that is used. So we can see this is a transect that we have used. And we can see that there are interruption on a belt line. So it will be interrupted belt transect. Biodiversity can be assessed at a number of different levels. Identify the levels of biodiversity that were assessed by this sampling technique that were not assessed by this sampling technique. On the fourth point here, we can see they have taken photograph and analyzed the photograph to get the percentage cover of the seaweed, percentage cover of invertebrates such as mussels and sea snails. So, in terms of identify the levels of biodiversity that were assessed by this sampling technique would be abundance or percentage cover of selected species. The levels of biodiversity that were not assessed by this sampling technique are range of different ecosystem or the genetic variation within each species. Table 2.1 shows the percentage change in abundance of some of the seaweeds, invertebrates, and birds found on the beaches of Hawadex Island. We can see seaweeds percentage change from 2008 to 2013 plus 2. So there is an increase, and percentage change from 2008 to 2019 is plus 49. So we can see then the increase becomes really high. However, the mussels, Mytilus, decreased from 2008 to 2013 by 94% and then decreased up until 2019 by 99%. The same happens with the sea snail, Litorina. It decreased up to minus 91%. Black oyster, Hematopus bachmani, increased up to 400% from 2008 to 2013 and then increased to 800% by 2019. The glaucous winged gull increased by 126% by 2013 and by 191% by 2019. Use the table 2.1, state the genus name of one organism that has increased in abundance. So we can see the most increase or highest increase in abundance is black oyster catcher, and the scientific name is Hematopus bachmani. First part of the name is the name of the genus. The second part is the name of the species in a binomial naming system. So it will be Hematopus. Seaweed were once thought to be plants but now classified in the kingdom of Protoctista. Outline the features of kingdom Protoctista that are shown by seaweeds. So we can write Protoctista have eukaryotic cells, which is similar to that of seaweeds. And seaweeds are photosynthetic, which is same with Protoctista. Protoctista plants are photosynthetic. And some additional points that we can also write is that they are aquatic. They don't contain any vascular tissue within them, which are features of kingdom Protoctista. To evaluate the success of Hawadex Island restoration project, the ecologist obtained data from other islands near to the Hawadex Island. Some islands had never been occupied by rats or they are rat-free islands and had a healthy ecosystem. Some islands were still occupied by rats. Rat-occupied islands had a disrupted ecosystem. So we can see the presence of rat causes disrupted ecosystem, and the absence of rat, the rat-free island, the absence creates a healthy ecosystem. Table 2.2 shows the percentage differences in the abundance of organism in rat-free islands compared with rat-occupied islands. So seaweeds, percentage difference in rat-free islands compared to rat-occupied. We can see there are a greater amount of seaweed, lesser amount of mussels and sea snail, and much higher amount of black oyster catcher and glaucous winged gull. With reference to table 2.1 and 2.2, suggest and explain how removing rat has restored the ecosystem on Hawadex Island. From the table here, we can see decreasing the amount of rats increases the amount of oyster catcher and glaucous winged gull, which are birds. So birds, their eggs are not eaten anymore. Their eggs or chicks are not eaten anymore, or less eaten, are no longer eaten by rats. However, the mussels in sea snail decreases because birds feed on them, and the amount of seaweed increases because there are fewer mussels or sea snails to eat them. So now, with reference to table 2.1, even in table 2.1, we saw that an absence of the rat had a general increase on the seaweed and the bird population. And we saw that there was a general decrease in mussel population and snail population. So the answer can be written in this way. Rats are no longer eating bird eggs and chicks, which increases the bird's population. Mussel, snail decreases because they are now eaten readily by the birds. Seaweeds increases because there are fewer mussels and snails feeding on them. Hawadex Island has changed to match into a rat-free island, or we can say rats are invasive species / predators. Oryza is a genus of grass plant that includes the rice. Oryza sativa, a food crop. Farmers flood fields of rice because this encourages faster growth and higher yields. An adaptation of rice plant that allows them to grow in water is the development of parenchyma. State the function of parenchyma. An parenchyma is a spongy tissue that forms air spaces or air channel in the leaves and stems and roots. So the answer can be written, parenchyma supplies oxygen to the submerged cells / tissues, and this aids aerobic respiration for those portions. State one other way in which the roots of rice are adapted to being submerged in water. Rice plant, typically when they are submerged in water, they can do anaerobic respiration for a prolonged period of time. So the answer can be written as follows. Rice plants are adapted to anaerobic respiration, or we can say rice plants have high tolerance to alcohol. Another adaptation in some varieties of rice is the fast growth of stems. Describe how selective breeding could produce varieties of rice with fast growing stems. Fast growth of stems is mainly due to the response of flooding. Selective breeding is the process where a human selects their favorable characteristics. Rice plants with fast-growing stems can be crossed with fast-growing stem rice plant. Then the offspring are chosen, and then they are bred. Then the offsprings are bred with fast-growing stems. Again, the process of crossing and selection then should be repeated over many generations to achieve the desired fast-growing stem rice. Auxin is a plant growth hormone that affects the growth of rice stems. Explain how auxin affects the growth of rice stems. Now, auxin is produced by the apical bud of a plant, and it is typically needed for cell elongation. So the answer can be written, auxin increases the stem height by cell elongation. At first, hydrogen ions are pumped into the cell walls, which break the hydrogen bond links between the cellulose molecules. Now you can mention the action of expansins over here as well. Then water enters the cell by osmosis and the cells elongate. So guys, this is a 3-mark answer. Any 3 of these points will give you the full marks. Just choose and write a particular sensible answer. Start from any point and finish whatever 3 points are done, and you will get your 3 marks. O. rufipogon and O. nivara are 2 species of wild rice. So O. rufipogon grows in places where water is always available. O. nivara grows within the same geographical range as O. rufipogon. The habitat of O. nivara can lack water for part of the year. The 2 species flower at different times of the year. These two rice species may have evolved by sympatric speciation. Explain how O. rufipagan and O. nivara may have evolved through sympatric speciation. So sympatric speciation is basically does not involve geographical isolation, occurs in the same plane. So the answer can be written, Onivara is adapted for drier conditions, and due to this and due to the different ecological isolation, Onivara had no gene flow with O. rufipagan, or we can say the reproductive isolation occurred due to polyploidy. Recombinant human proteins can be used to treat disease. Define the term recombinant DNA. So recombinant DNA is the DNA that is combined from two different organisms. From the 1920s until the 1970s, insulin obtained from the bodies of animals was used to treat diabetes. From the 1970s, recombinant human insulin was used instead. Explain the advantages of using recombinant human insulin to treat people with diabetes. So when person has diabetes, the person has inability to control their blood glucose concentration. So the advantages of recombinant human insulin can be written as follows. Recombinant human insulin is a protein that is made in bacteria. Now this is the opening statement. It does not provide any mark. So it can be made in larger quantities. It is cheap to make, or we can say it does not harm animals. Now the other points that we can say is that the protein is identical to human, so does not cause immune response, or we can say it can act more rapidly. Insulin is composed of two polypeptide chains, the A chain and the B chain. They are linked by disulfide bonds. Variations in amino acid sequences occur in the insulin molecule of different animals. In new versions of human insulin that have been engineered to control blood glucose concentration more effectively than normal recombinant human insulin, these new versions of human insulins are called analogs. So, the table shows the amino acid positions that this variation occurs in different animals and human analog insulin molecules. The dashes indicate a missing amino acid. So, we can see different amino acid positions and the type of insulin from human. We can see that in some organism, the position matches. Cats with diabetes can be successfully treated with insulin injections. Cat insulin is not available, but vet can choose from other types of insulin shown in table 4.1. Identify the type of insulin that is most suitable for treating cats. So to be suitable for treating cats, it must be close to identical to cat insulin. So cat consists of alanine, which matches with the cow in the A8 position, valine, which matches with the cow. Now, for the A18 position, there is no match. Then we can see asparagine, which matches with the cow. Asparagine also matches with the B3. Proline matches with the cow's proline at B28. And then lysine matches with the B29 lysine for the cow. And then alanine. So, in terms of most closest insulin that can be used in cat can be found from the cow. Suggest ways in which analog insulin molecules can be produced by genetic engineering techniques. So the answer can be written as follows. At first, normal human gene can be obtained from mRNA by using reverse transcriptase. Then we can use gene editing to alter the human gene and synthesize new gene sequence. Then we can put the new gene in bacteria to make insulin. The answer consists of 4 marking points. All of them are correct. You may forget any one, all right? And thus you can get full mark, all right, by writing 3 points. Any one of the 3 points will work for the 3 full marks. Information about the amino acids and nucleotide sequence is stored in computer databases. Outline the advantage of using databases of nucleotide sequence to investigate evolutionary relationship between species. So, storing nucleotide sequence in computer databases is known as bioinformatics. So the answer can be written in this way. The databases can be accessed from anywhere around the world online. This allows an easy comparison of multiple sequences whoever is working on the genetic information. Or we can say the comparison process becomes very fast because the comparison is done in a computer server. Or we can say it allows the counting of nucleotide differences, which would be very difficult for a human to do manually. Or we can say closely related species have few differences which can be investigated easily. The kidney is an important organ of homeostasis. One role of the kidney is osmoregulation. Figure 5.1 is a photomicrograph of a part of kidney nephron. We can see A representing the glomerulus and B representing the Bowman's capsule. The first portion asks us identify the structure labeled A and B, which we have done already. Describe how blood is filtered by the part of the kidney nephron shown in figure 5.1. So the answer can be written as follows. Ultrafiltration occurs in the glomerulus because the efferent arteriole is wider than the efferent arteriole, which creates a high pressure. The capillary endothelium has fenestrations or pores which allow small molecules to leave the glomerulus. Basement membrane forms selective barrier and podocyte supports the basement membrane. Now guys, we will only need 4 points to score the full marks. So you can write up until which allows the small molecules to leave the glomerulus into the Bowman's capsule. You will get the full mark just by writing those first 4 points. You may choose to write the other 2 points as well. However, do not be tempted to write more points, as making a mistake in the initial portion may lead to a deduction of a number, even if you write 5 points and one of the point is wrong. The cell surface membrane of kidney cells have receptors for many molecules, including glucagon and antidiuretic hormone ADH. Glucagon binds to G protein coupled receptors on kidney. So we can see glucagon binding with G protein coupled receptors. So this is G protein coupled receptors. These are alright. So this is the receptor. This is the receptor that is coupled with G protein. The binding of the glucagon to kidney cells activates cell signaling pathway that is similar to the cell signaling pathway activated when glucagon binds to liver cell. So as we can see, glucagon binding to the receptor coupled with G protein activates the G protein, and this G protein then stimulates a enzyme which kinase enzyme which turns ATP into molecule D, so which is cyclic AMP. So C is adenyl cyclase and D is cyclic AMP, and C would be adenyl cyclase. Figure 5.2 is an outline of cell signaling pathway when glucagon binds to kidney cells. Name the molecules labeled C and D. So as said earlier, C is adenyl cyclase and D is cyclic AMP or CAMP. Syndrome of inappropriate antidiuresis is a condition that affects osmoregulation in the kidney. Figure 5.3 shows how sodium ion concentration in the blood affects the ADH concentration in the blood in people with normal homeostasis and people with one type of SIAD known as type C SIAD. ADH concentration in the blood represented with picogram per cm cube. We can see sodium ion concentration in the blood in millimole per dm cube. Person with type C after 130 ADH concentration with type C SIAD. ADH concentration skyrockets, and the gray region is equals to normal range of ADH concentration. So after 140 it should increase. With type C SIAD, it increases right after 130, starts rapid increasing right after 130, whereas a normal person usually has an increase at or after 138, we can say, or 138, 139, something. Okay, now the question says describe the result shown in figure 5.3 and explain the effect of type C SIAD on osmoregulation. Our simple analysis will be, in both cases, increase in sodium concentration increases ADH concentration. So we can say a person with type C SIAD has increase at a lower concentration of sodium. So ADH increases at lower concentration of sodium. So this will be our description. Now we can explain what happens due to increase in sodium concentration and the effect of ADH concentration increase. So the answer can be written as follows. In both situations, as the sodium concentration increases, ADH concentration increases after a certain point. However, in type C, ADH rise occur at a lower concentration of sodium ion. Increase in ADH means more aquaporins and a greater permeability to water in the collecting duct. So in person with type C SiAD, more water is reabsorbed at the collecting duct into the blood. Domestic rabbits vary in the length and color of their fur. Figure 6.1 shows a domestic rabbit with short fur and a fur color pattern called Himalayan. Two genes that determine the length and the color of the fur of the rabbits occur at capital A/small a locus and capital B small bh and B locus. These two genes loci are on separate chromosomes. The allele A results in short fur. So capital A results in short fur. The allele small a results in long fur. Capital A is dominant to small a. So we can see in the diagram, first of all, this is a Himalayan and we can see short hair. So we can see short hair here in the rabbit and we can see Himalayan pattern. So a Himalayan pattern is when animals have black in their extremities and the rest of the body is white. So since there are three possibilities from the short fur, so we can have capital A, small a and small a, small a. Only the small a will result in long fur and these two will result in short fur, because short fur is dominant to long fur. The capital B results in black fur all over the body. The small bh results in black fur on the nose areas, paws and tails of the rabbit and white fur on the rest of the body, which is a Himalayan pattern. The allele B, small b, results in white fur all over the body, which produces albino. Capital B is dominant to BH and BH is dominant to B. So we can see this can give rise to many different patterns, one of them being having capital B and the other one either capital B or a small b or BH, any one of them, it can result in black fur because B is the dominant, okay, in all of the body. If small bh and small bh or small bh and B present, this will result in Himalayan pattern. All right, and only if small b and small b present, then we will get albino, because albino is recessive to all of them. Okay, now the question says list 4 possible genotypes of the rabbit shown in figure 6.1. So rabbit shown in figure 6.1 has short hair and has a Himalayan pattern. So to have a short hair, the first thing the rabbit needs to have is it needs to have capital A. So it can be both capital A, it can be one capital A with a small a as well. So with both capital A, now to have the Himalayan pattern, it must have either BH and BH or BH and a small b. So this also gives rise to second set. The second set will be either they can be both capital A with a small BH and a B, and then capital B and a small a with small bh and a bh. All right, so now just to make it more clear, let me switch this particular pattern from here to here. That makes much more uniform sense here. So as you can see, in both cases they are black, Himalayan black, actually not black as in Himalayan, and they are short hair. In all cases they are Himalayan and short hair. All right, because BH is dominant to small b and capital A representing short hair. Okay, one phenotype of rabbit always breeds true. A true breed is, all right, a true breed is a pure breeding animal. Okay, pure breeding animal, meaning that they are homozygous. They are homozygous. Now, this means that when it is mated with a rabbit that looks the same as itself, all the offspring looks the same as the parents. Describe the phenotype of the rabbit that breeds true. Okay, only true breeds can only be homozygous recessive. only homozygous recessive can be true breed. An explanation, I will give it to you, but let's look into the phenotype first of all. No, let's look into the genotype of it. So the genotype for homozygous for the hair length, for the fur length is small a, giving long fur. All right, and for the albino part, it will be small b. So it will have long fur and albino. Will have long fur and it will be albino. Now, the reason why a dominant character cannot be determined whether it is homozygous or heterozygous is that even if one dominant character is present, the other one being unknown, meaning it can be capital A, and capital A and a small a, yet they will produce the same color, same short hair. So it will be very difficult to determine, all right, whether they are heterozygous or homozygous. A rabbit with long black fur all over the body that was homozygous at both loci was crossed. So it had long, meaning it has small a. It had black fur, which is homozygous at both loci, meaning it had capital B and capital B. And then it was crossed with a rabbit with short and white fur that was homozygous at both loci. So short and white fur. So short and white fur can be described as short is capital A because it is homozygous, and white fur will be small b because it is albino. Okay, the F1 offspring had short black fur. So F1 offspring had short black fur. So it was short. So one of them is capital A, the other one obviously will be from one of the parent, which is small a, and black fur, so one of the character. So it will be completely heterozygous in both locations. These F1 rabbits were mated together to become the parents of the F2 generation. Draw a genetic diagram to predict the F2 offspring genotype and the ratio of F2 offspring phenotype. So first of all, F1 genotypes. Now we can already determine the F1 genotype, which is capital A, capital B, small b, and both parents will be capital A, capital B, small b. The gametes that they produce will be a combination of capital A, capital B, a capital A and a small b, a small a, capital B, and a small b, whereas the gamete on the other side would be also the same. So let's just copy and paste it. So both gametes are just the same. Okay. Now the F2 genotypes. Now the F2 genotypes that is going to be produced, well, it requires a diagram. Now to do the ratio of the F2 offspring phenotypes, first of all, we'll have to write down the possible phenotypes. So the possible phenotypes can be black with short. So we can say black short ratio with white short ratio with black long with white long. So at first we can do the black short. So first of all, to have black color, there must be a capital B present. And to have short, there must be a capital A present at any point. So it's very easy to determine. So all the yellow labeled ones, these are they contain capital A with capital B, at least one of them. So capital A with capital B. Now, if we do the counting, this comes 1, 2. So this comes at a ratio of 9. So 9 ratio with, now we will do white short. To do white short, short has to have a capital A, and to have white, it has to have small b. So this time we're doing the labeling with the red. All right. So it will be 1, 2, 3. So 3 of them. There will be 3 of them only. Now we will do for the black long. To have the black, there must be a capital B, and to have long, there must be small a's. So there are 3 instances that occurs. So that will also be a ratio of 3, and then only one left, which is white long. A rabbit breeder performed multiple crosses of the type described in B. This gave enough data to test the prediction that the genes for fur length and fur color show independent assortment. Explain why 2 genes assort independently. Independent assortment is when the genes are on separate chromosomes and they are not linked. So, in terms of explanation, why two genes assort independently, we can say because they are on separate chromosome. Two genes assort independently because they are on separate chromosomes. So, each pair of homologous chromosome orients itself separately. This gives four new combinations of alleles, and parental allele combinations are not preserved in these new combinations. The rabbit breeder placed the results in a table and started to calculate the chi-square value. Table 6.1 shows the results and some of the calculations made. Calculate the expected numbers and write them in the shaded column in table 6.1. Use the formula provided and the figures in table 6.1 to calculate the chi-square value. So the chi-square is equals to sum of observed minus expected whole square divided by expected. So observed minus expected whole square. Now we have to add all of those values. So we have to add these particular values to get the total chi-square value. So 0.14a plus 0.444 plus 0.111 plus 0.333. This gives a total value of 1.036. So chi-square has a total value of 1.036. Amino acids are the monomers that are used to produce proteins in organisms. Amino acids also have other non-protein roles in the body. Figure 7.1 shows the structures of 5 amino acids with varying numbers and arrangement of carbon atoms. We can see here asparagine, leucine, methionine, proline, and serine. In the liver, one of these amino acids can be converted to pyruvate and one of these amino acids can be converted to oxaloacetate. Suggest which of the amino acids shown in figure 7.1 would be most directly converted to pyruvate and oxaloacetate. Amino acid converted to pyruvate. Now, pyruvate consists of 3 carbons. Now, the ones that can turn into pyruvate and consist of 3 carbons is serine, as you can see here. The other ones are not a possible choice because this one has a combination of 5 carbon. This one is a combination of 4 carbon. This one is a combination of 6 carbon. And this one is a combination of 4 carbon. Now, one that can turn amino acid that can be converted into oxaloacetate. Now, oxaloacetate consists of 4 carbon. So, we have to look into the compound that contains 4 carbon. Now, the 4 carbon containing compound is asparagine. Another molecule that consists of 4 carbon, which is methionine, but since it also contains 1 extra carbon, so we can leave that one out in terms of possibility. The amino acid glycine can act as a neurotransmitter. A glycinergic synapse is shown in figure 7.2. We can see the mitochondria is providing the energy, the ATP, for the process of exocytosis. When calcium ions enter, the exocytosis of glycine occurs. Glycine sits in the receptor, which allows the influx of chloride ion. The glycinergic synapse and cholinergic synapse use different neurotransmitter and different postsynaptic receptors. Describe differences between glycinergic synapse shown in figure 7.2 and cholinergic synapse. Now we know that in cholinergic synapse we use a neurotransmitter, all right, and the neurotransmitter is acetylcholine, ACH, whereas in glycinergic synapse we are using glycine as our neurotransmitter. So the chemical that we are releasing here is glycine. So it's different as a neurotransmitter compared to cholinergic. In cholinergic synapse, we use sodium ions in the postsynaptic membrane for depolarization. And in cholinergic synapse, it is the influx of chloride ion that leads to depolarization. So the answer can be written as follows. Chloride ions, not sodium ions, are entering the postsynaptic neuron in a glycinergic synapse, and glycine is not broken down, but acetylcholine is hydrolyzed in synaptic cleft. The binding of glycine to receptors, as shown in figure 7.2, makes an action potential less likely to occur in the postsynaptic neuron. Suggest why an action potential is less likely to occur after the binding of glycine to the receptors. So once the glycine is binding to the receptor, it causes an influx of chloride ions. Chloride ions are responsible for depolarization. Chloride ions are responsible for repolarization. repolarization. Since repolarization occurs, it becomes difficult to depolarize. Chloride ions decreases the resting potential difference, so it needs more depolarization to reach the threshold. And as an added point, we can also say glycine acts as an inhibitory. Only 2 points will be needed to score the mark, so write any one of the two, any one of the three. Neurons need to maintain a resting potential before an action potential can occur. Describe how a neuron maintains a resting potential. Resting potential is maintained by maintaining a potential of minus 70 millivolt. Resting potential is maintained by the sodium-potassium pump. Sodium ion is pumped out and potassium ion is pumped in. 3 sodium ions are pumped out and 2 potassium ions are pumped in by 1 ATP. However, some potassium ions can diffuse out of the membrane, which makes inside of the axon more negative relative to the outside of the membrane. Figure 8.1 shows a cell from the testes of a locust at the late prophase 1 stage of meiosis. Explain how the behavior of the chromosome in prophase 1 of meiosis results in the appearance shown in figure 8.1. From the diagram, we can see that the nuclear envelope disappeared. Chromosomes are condensed. The chromosomes condenses. Homologous chromosomes pairs up and crossing over occurs between non-sister chromatids. Finally, we can add a point. Chromosomes stay joined at the crossover points. Now, the reason the answer is like this because our question says that we have to give the behavior of the chromosomes in prophase 1. So, we just have to mention the behavior of the chromosome only. Many factors affect the rate of cellular respiration. Pyruvate carboxylase (PC) deficiency is a rare disease. The rate of respiration in the cells of a person with PC deficiency is much lower than normal. The passage describes the occurrence of PC deficiency in human population. Complete the passage by using the most appropriate scientific term. PC deficiency affects 1 in 250,000 people. A person either has the disease or does not. So there are no intermediate forms of the disease. These can indicate that the disease is caused by one single gene. PC deficiency is caused by a genetic mutation in one gene. Whether a person develops the disease is not influenced by, so it is not influenced by environmental factors because it is caused by one single gene. So the word should be environmental. The distribution of PC deficiency in human population shows, since we can see there are no intermediates, and no intermediate means that this is a discontinuous variation. Coenzymes are important in aerobic respiration. Outline the role of named coenzyme in aerobic respiration. Coenzymes in aerobic respiration includes coenzyme A, and we can talk about NAD or FAD. Since the question has 5 marks, so we can write the description as follows. Coenzyme A binds to acetyl group. This joins the link reaction and Krebs cycle. NAD/FAD transports hydrogen ion and electron and acts as a coenzyme for the enzyme dehydrogenase. Thus, it transports hydrogen ion and electron to the EDC for oxidative phosphorylation. NAD accepts hydrogen in glycolysis and link reaction and Krebs cycle. Sweet peas are garden plants that vary in height. Tall sweet peas grow to 200 cm in height. Dwarf sweet peas grow to 30 cm in height. Tall sweet peas contain a dominant LE allele. Dwarf sweet peas are homozygous for the recessive LE allele. Explain how small le genotype results in the dwarf phenotype in sweet peas with reference to the effect of small le on enzyme synthesis, hormone production, and the expression of gene affecting the plant growth. So, the small le genotype codes for the mRNA, which then is decoded by the ribosome for the enzyme synthesis. And then the enzyme is then converted to hormone by the action of smooth endoplasmic reticulum. And once the gene is expressed, the hormone is produced. The hormone brings about the plant growth response. The small le allele codes for a less functional enzyme. So as a result, in the enzyme, alanine is replaced with 309 at the active site. This results in inactive gibberellin, or the inactive gibberellin is not converted to active gibberellin by the enzyme. As a result, less gibberellin is available for binding with the receptor and forming DELA complex. And DELA is not broken down as a result. So DELA stays bound to the transcription factor, and transcription factor is unable to bind with the promoter. So the growth genes are not expressed. Guys, that's all for this particular question paper. Thank you for joining this particular video. And guys, recent question paper videos like this will be uploaded regularly. Subscribe to the channel so that the channel can grow. And we feel a little bit of encouragement to keep uploading more question paper. Thank you guys. See you in the next video. Bye-bye.